Hello everyone. Today we are going to discuss about the voltage clamp and patch clamp technique. Patch clamp technique. So if we will go back into the history, Hawking and Huxley revealed the ion channel event. They revealed the ion channel event, how the ions, how there is the change in the current during the action potential, and this change were recorded by using the voltage clamp technique, and both were honored with the Nobel Prize in Physiology and Medicine in 1963. During this period, voltage clamp could only be applied to big cells only, means they were not able to utilize this technique to the smaller cell. Then another two scientists came into existence, Nihir and Sackman, in the late 70s, which which modified the voltage clamp technique, or we can say they advanced this voltage clamp technique. And then in the first time of the history, they were able to determine the single channel current across a membrane, and the whole experiment were carried out in frog skeleton muscles, where they were using patch clamp technique. And again, they both were awarded with the Nobel Prize in Physiology and Medicine in 1991. So, if I will talk about the principle of voltage clamp, it's like we are not able to study the behavior of sodium and potassium channel, because the conductance through these channels changes with action potential. As the action potential propagates, their conductance also get changes, and their voltage also get changes. But with the help of this voltage clamp technique, we were able to study these ion channels. Voltage clamp is a technique which is used by electrophysiologists to measure the ion current through these membrane when the cell is in the excited when the cells are in the excited state, like the nerve cells or the cardiac cells or the neurons, while holding membrane voltage at a desired constant level. So whenever we have to go for studying the ion current, when the cells are in the excited state, that time we have to hold the voltage at a desired level. The basic voltage clamp measures the membrane voltage, then changes the voltage at to a desired value by adding the necessary current. This clamp By this clamp, the cell membrane at a required constant voltage allow the voltage clamp to record what current we delivered. Whatever current we are delivering, this voltage clamp is measuring that current, because current applied to the cell must be the current going across the membrane at a set voltage. The recorded current indicates how the cell react the change in the membrane potential. As you can see in the diagram, this was one of the experiment or the technique how this voltage clamp techniques were introduced. So we are having Petri plate. In Petri plate we have taken squid axon, in which on its left hand side we have kept recording electron, and on its right side we have kept current passing electron. So current is passing through this electrode only, and the whole system is kept in the saline solution, in which we have kept our reference electrode as well. So one internal electrode measures the membrane potential, which is kept inside the squid axon and is connected to the voltage clamp amplifier. As you can see in the diagram, this recording electrode is connected to the voltage clamp amplifier. This voltage clamp amplifier comprises membrane potential to a desired potential. When this membrane potential is different from the command potential, the clamp which is the clamp amplifier which is there injects current into the axon through the second electrode. This feedback arrangement causes the membrane potential to become as same as the command potential. So the current flowing back into the axon and thus crosses its membrane and can be measured by the voltage clamp. This is how basic technique how this patch clamp technique were used. So what is happening in the patch clamp technique? We can say it's a very powerful technique and flexible technique too, which can be used for to study the biological properties of the membrane. It allows recording of macroscopic whole cell or microscopic single channel current flowing across the membrane. By this technique, we are able to see the current flowing through small channels, like example a sodium potassium pump, like that. Different configuration of patch clamp techniques have been developed. So different patch clamp techniques are there, and these different patch clamps are used for different types of cells. Among these configurations, cell attachment patch clamp technique is the most useful technique, which is used to study the single channel, which is used to how current is flowing through the single channel. If we talk about the principle of the patch clamp technique, it is like that: a glass pipette with a very small opening or tiny opening containing the electrolyte solution is firmly sealed onto the neural membrane. So pipette is kept on the neural membrane. This pipette is having the electrolyte solution in it, or other membranes we can use, and thus isolate the membrane patch electrically. After application of small amount of suction to the back of the pipette, the formation of a high resistant seal in the gigaohm range occur. This tight sealing isolates the membrane patch electrically, which means that the all ions fluxing the membrane patch flow into the pipette and are recorded by an electrode which is connected to a highly sensitive electronic amplifier, as you can see in the diagram. To prevent the alteration in the membrane potential, a compensating current that resembles the current flowing through the membrane is generated by this amplifier as a feedback mechanism. The membrane potential of a cell is measured and compared to the command potential. If there is difference between the command potential and the measured, a current is injected then only. This compensation current will be recorded and allow the conclusion about the membrane conductance. So we are having different configurations of the patch clamp techniques. These are cell attached or on cell patch, inside out patch, whole cell recording, outside out patch, perforated patch, loose patch, and automatic patch clamping. This is how we are having different types and configuration of the patch clamp technique. So here in this diagram what we have in the first figure, we are having cell attached recording where we have kept our recorder pipette. So by giving mild suction, this tight due to this mild suction, this pipette gets tightly attached onto the membrane, as you can see the second one. And then when we are taking back inside out recording, when we have to go for, or we are going this taking out or to expose to the air, this cytoplasmic domain is accessible, by which they are able to measure the current or the changes which are occurring in these small channels, like here. Now in the second case, what we are doing, whole cell recording. Here we are using strong pulse of suction. So what is happening there, cytoplasm is continuous with the pipette interior, as you can see the diagram. Then first we have to make the tight contact between the membrane and the pipette. Then the strong pulse of suction is applied onto the membrane. Then this cytoplasm is continuous in the pipette interior. Then we have to retract the pipette. We have to take pipette from the membrane. Then this allow us to go to record the outside out recording. So what is happening in the last end of the membrane's anneal, as you can see here, this is the extracellular domain accessible. This is how these small channels or ion channels are separated from the membrane and recorded by these pipettes. Or we can say, if we have to go for the applications of the patch clamp technique, we can use this patch clamp technique for the evaluation of anti-arithmetic agents, or we can use patch clamp technique for identification of multi types of calcium channels. When the cells are in excited state, that time there is action potential. So by these patch clamp techniques, we are able to identify what multi types of calcium channels are involved. Or we can use this to measure the effect of potassium channel openers. We can use this for the study of the sodium channels, as we know we are having different channels present on the membranes. We can use to investigate a wide range of electrophysiological properties of the membrane. Electrophysiological properties of the membrane. We can also use this technique for the measurement of cell membrane conductance. So these are the most important applications of the patch clamp technique. Now we are having limitations too, as well, as it's one of the very expensive technique, so it's not available every time everywhere. In this, like we have seen while discussing this technique, we have seen that it is a time consuming technique, as well as we have to fix our pipette onto the membrane. There should be proper fixing of the membrane. If it is not, then we can't use this technique. There are chances of membrane distortion. Number of samples are required, and we have to proceed, or we have to use this technique, or we have to apply this technique many and many times, so that we will be having proper results. And to carry out this technique, what we have to do, we need a skilled person, so that all the ion channels which are present on the membrane should get inserted into the pipettes. Then only we will be able to go for the recording of the cells. This was how we are using these techniques to study the membrane potential, or to study the current which is flowing through these ion channels. Thank you.